My name is Anthony Burnetti. I work in the Ratchet laboratory here at Georgia Tech, and we are interested in the origin of animals, and I will be talking about something that's not actually what we do in the lab, but just some analysis I have been thinking about regarding the role of evolutionary priority effects and evolutionary ratchets in the evolution of the Holozoa. So there's I've shown this slide at a bunch of different talks at this point. It seems like sort of an organizing principle of the problems I'm interested in is that there's this endless argument in evolutionary biology about whether major innovations and major evolutionary transitions are predictable and easy to evolve or contingent and hard. There's this wonderful book, 1970s, with about people look at biochemistry and just say, oh my God, this is so strange. We finally know we are alone in the universe. And other people look at the history of life and they say, okay, this makes sense. This makes sense. All this is fairly orderly. I don't see why this shouldn't happen all the time. And everybody is looking at the same thing. And so what this says to me is that people aren't doing this systematically. You're using quick first impressions, and you need to do this more systematically and try to figure out what's actually going on. So, like I said, I work in the Ratchet lab. We are interested in the origins of multicellularity. These things, these large macroscopic organisms where a single subunit is repeated over and over again to make a much more interesting, larger whole, these define the world today. And multicellularity in some form has appeared 25 plus times over the history of life on Earth, and 6 or so times that are particularly complicated and interesting. Got your animals, your fungi, your plants, your various kinds of algae. And some of them seem to be fairly similar to each other. Down on the bottom there, you got your brown algae and your red algae. Are they really all that ecologically different? Arguably not. You do have some, though, like, say, animals that are fairly unique. There's nothing else quite like animals on Earth. So we like to examine this in the lab using using laboratory evolution. We evolve microbes into very simple multi-celled forms in the lab. But that only gets you so far. There really is an important role for looking at the actual evolutionary history of actual organisms as they've occurred on Earth and trying to figure out what that tells you about their history. So to understand the origin of these guys, you should examine their closest relatives. And the closest relatives that are microbes to animals are the holozoa. This is a set of multiple different organisms. You've got your choanoflagellates, who are the closest relatives. You've got your ichthyosporia that are a little more closely related. You get Phyllis, all these different things. There's a couple of them. And they can be unicellular, or they can live in simple multicellular groups. These are some pictures of colonial choanoflagellates that swim around, and some of them even change shape in response to light. And some of them have interesting multiple life stages in cell types. They can have complicated life cycles. And down here, just a quick little video going, this is one of those ichthyosporia I told you about. This is a single large cell that will grow, and then all of a sudden it will shatter into all these little cells. It cellularizes in a way that's weirdly similar to embryogenesis, and then everything goes away. So, and of course, some of these things resemble things you see in animals. Choanoflagellates actually resemble some cells you see in sponges really closely. And Holozoa as a whole, people often call them potentiated for the evolution of complex multicellularity. Like I said, they can have these really complex life cycles by which they have lots of different cell types that behave differently. Some people say, well, that's sort of pre-adaptation for differentiation. Instead of separating cell types in time, you separate them in space. These guys have extracellular matrix, adhesion proteins, signaling proteins, all kinds of things. You can really see how you might say that having all of that makes it easier to evolve multicellularity, but that raises an interesting issue, and that's if a lineage has to be potentiated for a long time, if it has to get all of these in a unicellular or tiny colonial context, that would indicate that a transition to a complex multicellular organism would be more difficult or contingent. But I would like to raise and amplify an alternative hypothesis. And that is, are these unicellular holozoa really potentiated, or are they unicellular revertants? This is a thing that happens over evolutionary time sometimes. This picture here, this is not holozoa. I've got a filamentous fungus on the left and Saccharomyces cerevisiae, beer yeast on the right. These are single-celled fungi that have lost all capacity to live as groups because they moved into a different ecology, and that became what was favorable. So is it really all that difficult to think about other possibilities in other lineages? And especially with these holozoa, they've got a lot of interesting, odd features that people like to talk about being potentiated, but I think they could equally well be explained by reversion. Instead of a lineage developing lots of traits that then get elaborated into multicellular traits, you could have a simple multicellular organism that gets all these traits for that purpose, and then those get exapted into unicellular lifestyles. This gets rid of a lot of the contingency that would be behind the evolution of animals, and it reverses your evolution of differentiation idea. Instead of specialization across time becoming across space, it becomes specialization across space becoming across time. And this is, I think, very, very possible because there's two things you need to understand why this, I think, is somewhat likely. And one is that major evolutionary transitions and innovations are not single events. They do not leap into existence suddenly. There's large numbers of intermediates. In our lab, we see a really simple toy version of this. Ozan is a fantastic postdoc in our lab whose work, honestly, all of us are going to be building on and dependent on for so long. He has done a beautiful 2-year-long long-term evolution experiment where we evolve these simple multicells into enormous multicells. And along the way, there's many, many small changes. There is not one single genetic change that causes all of this. And there are other examples of this in actual history. And so what you would actually see is more along in terms of major evolutionary innovation. Instead of one event, you have lots and lots of events where something invents something new, competitive exclusion drives competitors to extinction, things can diversify, the diversity goes away when somebody gets a really major invention that gives an advantage. There's many steps along this pathway. And secondly, there's something called an evolutionary ratchet that can occur. Now let's explain this real quick. Early after a major evolutionary transition or innovation, there's only a few genetic changes that constitute it, and so there's reversion would require a few changes and revertants are fit. But later on, as they get more and more canalized into living that way, more changes become required to revert that phenotype, and the revertants become less and less fit. And this is another thing we see in our own lab. This is a picture of one of our multicellular yeast groups that we evolve in our laboratory. The red cells are dead. These cells have actually actively killed themselves. And they do that because when they're growing in a multicellular context, having 5% of your cells kill themselves is actually a good way to make sure that propagules of the proper size fall off and found the next generation. But if we artificially revert these guys back to unicellularity, they are less fit because the cells are just randomly dying some of the time. So these ratchets can lock you into a new state. So I would like to propose this model of the holozoa where all these unicellular or simple colonial things are sort of these straggler lineages. And this could come about if the evolution of multicellularity is not instantaneous. You start off with a multicellularization, you have simple multicells, it diversifies, and eventually one lineage ratchets into multicellularity and gets major advantages and others can't compete with it. And the only surviving lineages from down along its stem that you would see would be those that revert to unicellularity early from a simple multicell. And these would retain signatures of a simple multicellular ancestor, but not retain multicellularity itself. And this has implications, model has implications for many innovations and transitions. I think that if this is true, innovation followed by diversification can appear like contingent potentiation when reversion is initially very possible. And the existence of these revertant lineages depends on the timing of the evolutionary ratchets that prevent reversion in some lineages. And it would also suggest, I think, that long stem groups where you have one group that is very different from its sister groups, that has many, many major inventions along the way, that would be an indication of early ratcheting and very strong competitive exclusion and evolutionary priority effects. Now, and I would just like to say, I think there's other possible examples of this, and I would just like to plug one idea. I wonder if this could also be relevant to something as fundamental as eukaryogenesis. On the right here, you have a tree of the Archaea. And in colors, you've got these brand new Archaea people are so excited about recently called the Asgard Archaea. These Archaea have all kinds of eukaryote signature genes involved in our key eukaryotic processes, all kinds of membrane remodeling, all kinds of cytoskeletal protein, elaborate ribosomes that are elaborate in the same way that eukaryotes are, and the eukaryotes root within them. So people are saying, oh, these are the key to understanding eukaryotes. And then somebody finally gets a picture of one of them. They're a little underwhelming. This is a long story behind finally this is the only Asgard Archaea we have a picture of. It doesn't have complicated internal architecture. It doesn't have obvious cytoskeleton. It's got a few interesting tentacles, but overall, some people look at this and say, is that it? I wonder if they could be degenerate proto-eukaryotes. They retain these eukaryotic signatures, but maybe they were using them for something more akin to a proto-eukaryote, but they've reverted to an archaeal lifestyle when the main eukaryotic lineage got really good at what it was doing and excluded them from that niche via competitive exclusion. And eukaryotes, they have an enormously long stem along the evolutionary tree. And I wonder if this is, once they've sort of ratcheted in where they can no longer throw off lineages into previous ecological niches, this could be the result of many waves of competitive exclusion where they invent something big and important along the way. So in conclusion, I think that it's important to realize that ecological interactions and hopping back and forth between ecological niches can make parsimony a poor assumption in an evolutionary tree, potentially obscuring true histories. Evolution does not only run one way. I think that combinations of these evolutionary ratchets, competitive exclusion, and priority effects can make innovations potentially appear much more contingent than they actually are. And I would love anybody's input on how to test this hypothesis. I've been thinking about what simpler multicellular holozoa might look like in the fossil record. I have ideas for long-term evolution experiments in the lab where we actually take our multicellular yeast and make reversion a little bit easier and test under what circumstances reversion to unicellularity occurs. I also have some thoughts on modeling the topologies of evolutionary trees that you get in circumstances where mutations can change investment and return on different strategies, in addition to just fitness along a single strategy. With that, I want to thank everyone in the Ratcliffe lab, especially Ozan, postdoc whose work has inspired all of us in the lab so much, Will for letting me think about these odd things while I do my evolution experiments, and Ollie for constantly listening to me rant about these things. And